Lymphocyte and antibody response. We have mentioned before that one of the characteristics of the specific immune system is that it has memory. This means that the first time an antigen is encountered, the response is delayed. It may take 10 to 12 days before antibodies appear in the case of the antibody-mediated immunity. After the first response subsides, memory cells have been made that remembers the encounter with the antigen. After a period of time, if that same antigen is encountered again, the second response will be immediate and enhanced. So characteristics of the first response is class switching, affinity maturation, and formation of memory cells. As long as the antigen continues to be present, some B cells will continue to undergo changes that will enhance the immune response. So as long as the antigen is present, class switching, affinity maturation, and formation of memory cells will take place. The lag period that is seen during the first immune response, the first encounter to the antigen, lasts 10 to 12 days. This is the time needed for clonal selection and clonal expansion. This lag period of 10 to 12 days is the time that passes before antibody is detected in the blood. Again, as long as the antigen remains present, B cell proliferation continues. So the selected B cell will continue to clone itself, producing B cells, memory cells, and a slow steady increase of IgM, the first antibody that appears during the first immune response, will be seen. When the B cell is activated, its daughter cells will eventually become plasma cells that secrete IgM antibodies. IgM antibodies are thus the first antibodies that appeared. Notice that eventually some of the clones of the original B cell will begin to secrete IgG antibodies. After a period of time, as long as the antigen continues to be present, IgG antibodies will be the preferred antibodies secreted by plasma cells. This event is called class switching. You can see class switching in the graph right at the point where IgGs appeared. Also notice that the titers or concentration of IgGs will be greater than that of IgMs. The second event that happens during the first immune response is something called affinity maturation. As long as the antigen continues to be present, affinity maturation will allow for a better and better immune response. This means that the antibodies made will be more and more specific and will bind the antigen tighter. Essentially, the first clones created by the B cells do not bind the antigen as tightly as subsequent clones. The cells that have better affinity for the antigen will be stimulated to divide, and their offsprings will have even better affinity to the antigen. After a period of time, the more specific plasma cells will remain, and those will be the ones making antibodies at the end of the response. These antibodies will be IgGs, and it will be of a more specific kind. Again, the delay during the first immune response occurs due to expansion, clonal selection, and clonal expansion. The first antibody made is IgM. Shortly after, class switching takes place and plasma cells begin to secrete the more specific IgGs. As long as the antigen continues to be present, affinity maturation will occur, producing more specific IgG antibodies. Formation of memory cells. Memory cells are B cells. These are daughter cells of highly specific B cells selected during affinity maturation. So affinity maturation leads to the formation of memory cells. When memory cells are activated during the second immune response, they will immediately begin to produce very specific IgG antibodies. So IgG is the antibody of memory. Memory cells are long-lived cells. They circulate in the body for years, allowing for protection. Vaccines exploit the phenomena of immunological memory. So during the secondary immune response, memory cells are responsible for a quick and swift and enhanced reaction. Very quickly, memory cells differentiate into plasma cells, which produce large quantities of very specific antibodies. Affinity maturation has already taken place during the first response to the antigen. This is the second response to the same antigen. The response takes hours instead of days. Very quickly, IgMs will be made, but also IgGs, very high levels of highly specific IgGs will be produced during the secondary immune response. Vaccines represent the first response to the antigen, which will hopefully elicit formation of memory cells. If the same antigen is ever encountered again, the second response will be very swift. Within a matter of hours, memory cells will differentiate into plasma cells, which will produce very high levels, titers, of IgGs, very specific IgG antibodies. The patient will never even be aware of the enhanced immune response, which will take care of the threat swiftly and effectively.